the female flower, it is one of the branches coming out from the near the leaves. So this female flower, once the air move, so the pollen grains fall down and fertilize the female flowers. So they are on the same plant. The second type is called cross-pollination. At this moment, we found that the pollen grains are transported from the anther of one plant to the stigma of another plant. But they must be from the same species. So another time, pollination is the transferring of pollen grains from the anther to the stigma of the flower. Types of pollination, it is the first one, it is the self-pollination, in which the transferring of pollen grains from the anther to the stigma of the same flower, or to that of another flower of the same plant. Another type, the cross-pollination, this takes place by transporting or transferring the pollen grain from the anther to the stigma of another plant, but they must be from the same species. So pollination, it has two types, self-pollination or cross-pollination. In order that the cross-pollination takes place, there is some factors must be found in the flowers. First, the flowers are unisexual, so they have to be unisexual one. Second, that the sex organ or one of them, male or the female, mature before the other one. The height of the anther is lower than that of the stigma. So another time, the main factors of the cross-pollination, the first one, it is the flower are unisexual. The second factor, the sex organ of one sex becomes mature before those of the other one. The height of the anther is lower than that of the stigma. Then what are the ways of this cross-pollination? How the pollen grains are transferred from the anther to the stigma? This cross-pollination takes place. Sometimes they are carried by the air. Another type, they are carried by the wind, or they are carried by insect or by water, and in general sometimes they are carried by the man. Now the pollen grains are on the stigma. Once the pollen grain reaches the stigma, so at this moment pollination is fulfilled and finished. Then once you have a guest, you give him something, juice or something like that. Also, once the pollen grain reaches the stigma, so the stigma becomes very excited, so it give it sugary juice in order to help it to grow. Then the pollen grain begins to grow and form the pollen tube. The second step of pollination, it is fertilization. Now we are going to study fertilization process. The fertilization process is a process in which the male gamete combines with the female gamete to form zygote. The fertilization, it is a process in which the male gamete combines with the female gamete to form zygote. We knew from the first step that the pollen grain or the male part reaches the female part by the help of pollination, but it reaches the stigma only. How it will go inside until it reaches the ovary in order to combine with the ovule. At this moment, we found that this process takes place according to the following stages. First, the pollen grain, it begins to germinate according to the juices it takes in and form the pollen tube. This pollen tube germinates and forms the pollen tube. It contains two nuclei. We remember them from the last time. Yes, who can tell me? Yes, the generative one and the tube nucleus. So we have two nuclei, which are the tube nucleus and the generative nucleus. The generative nucleus and the tube nucleus grow. The tube nucleus pull the tube, pollen tube, until it reaches the micropyle. Very good. So the pollen tube penetrates the stigma and cross through the style till it reaches the micropyle of the ovule. Very good. So the micropyle here lets the pollen tube enter to it. Then the tube nucleus degenerates while the generative tube begins to divide mitotically into two male nuclei. So the pollen tube divides into two male nuclei. Each one of them fuses with the egg nucleus. One of them only fuses with the egg nucleus, forming the zygote 2N chromosome, which start to divide, forming the embryo 2N chromosome or diploid one. While the other one go to the polar nucleus and fuse with them, forming an endosperm nucleus 3N chromosome or triple fusion. So this type of fertilization, it is called double fertilization. Another time we are going to repeat this part because it is a very important one, and most of the questions comes in this part. So once the pollen grain reaches the stigma, it secretes a juicy substance which is absorbed by the pollen grain and germinate, forming the pollen tube. This pollen tube penetrates the stigma and crosses through the style and till it reaches the micropyle of the ovule inside the ovary, and enter from the micropyle into the ovary inside it. And at this moment the tube nucleus degenerate, while the generative nucleus divide into two by a mitotic division into two male nuclei. The first one fuses with the eggs, forming zygote, while the other one fuses with the egg and forms the zygote and start to divide, forming the embryo, while the other one fuses with two sac nuclei and form endosperm nucleus, which has 3N chromosome. This is called double fertilization. The double fertilization, it is the fertilization of egg nucleus by male generative nucleus to form the zygote. This is the first one. And the fusion of the male nucleus N with the two embryo sac nuclei, forming the endosperm nucleus 3N. At the same time, the endosperm nucleus is divided to form endosperm tissue. So the endosperm here, the 3N, it will divide and form the endosperm tissue, which act as a source of food for the early developing embryo. So that's what happens inside the ovary, and that's what we are calling the fertilization. And inside the fertilization, we call it another name, which is double fertilization. Why it is double fertilization? Because the generative nucleus divides into two. One of them fuses with the egg and forms the zygote. The second one fuses with the embryo sac nuclei and form the endosperm nucleus. Then the endosperm tissue is formed by the developing of this endosperm tissue. After that, what happens to the ovary and what happens to the organs we studied? This will lead us to the final point of the functions of the flower, which is, yes, the formation of fruit and seed. According to the presence of the endosperm tissue, the cells are classified into two main groups. According to this, we can divide the cells into two main groups. And especially the seeds, the endosperm tissues, the cells are classified, or the seeds are classified into two groups. First group, endospermic 1 or endospermic seeds. The second group is exondospermic seeds. The endospermic seeds, briefly, it contains endosperm. And the exondospermic seeds that doesn't contain endosperm. The first one, the endospermic seeds. The endospermic seed, they are seeds which contains its endosperm tissue, occupying a part of the seed outside the embryo, such as the monocote seeds. In monocote seeds, we found that the ovary and the ovule fuses together, forming single seeds fruit called grain, such as the maize and wheat. Another time, in order to understand this very well, endospermic seeds, they are seeds which contains endosperm tissue that occupy a part of the seed outside the embryo. So they are founding around the embryo. This is found in general in monocote plant or monocotyledonous plant. The seeds here in monocotyledonous plants, we found that the ovary and the ovule fuses together, forming what we call it the grain, a single seeded fruit, forming a single seeded fruit called grain, as in maize and wheat. In exondospermic seeds, they are the seeds which does not contain endosperm, because the embryo feed on the endospermic tissue during its development. So the food of the embryo is stored in the two cotyledons, the integuments of the ovule, harden and form the testa or the cover of the seed. Another time, exondospermic seeds. They are seeds which does not contain endosperm, because the embryo feed on the endospermic tissue during its development, and it stores the food into the two cotyledons. So this is what we call it seed. So the integuments or the cover of the ovule, they are hardened and form a testa which cover the seed from outside and to protect it. And this is such as the seed of bean and pea seeds. So the difference between the endospermic seed and the exondospermic seeds, that the endospermic seeds, the endosperm is found surrounding the embryo. But in that of the exendospermic seeds, the seeds, they are the endosperms here tissue. It is consumed during the development of the embryo, and the food, it is stored in outside the embryo into the two cotyledonous one. The integuments, they harden a little bit, and they cover from the outside the seed itself, so it protects it from the environmental conditions. Example for these exendospermic seeds, they are the bean and the pea seeds. Then what happens after the fertilization to the other parts of the flower? After fertilization, the calyx, the corolla, and androecium, the style, the stigma, they wither or wither and fall down. Only the ovary remains that store the food and change into fruit. This is referring to some hormones, or what for the plants we call them auxins, secreted by the ovary. So the ovary wall is transformed into the pericarp and the seed wall into the coat. Okay? Another time. After fertilization, the calyx, the corolla, and the androecium, the style, and the stigma, all of these parts of the flower fall down, they wilt and fall down. Only the ovary remains. It begins to store the food and swollen and change into the fruit one. This is helped by a certain types of hormones which is secreted by the ovary. The hormones type of the plant, they are called auxins. So the auxins secreted by the ovary, they give the chance for this ovary to swollen and store food. The ovary wall transformed into pericarp and seed wall into coat. The two synergid cells and the antipodal cells disappears or degenerate, while the micropyle remain to allow water to get into the seed during germination. So another time, if we look for this part, you are going to find, remember the synergid cells? Yes, the cells which are surrounding the egg cell. So these two will degenerate. Also the antipodal cells, which are found at the other pole of the ovule, they degenerate also, and only the micropyle remains in order to help the water to enter during germination of the seeds. The formation of the fruit and seeds. If we look to the fruits, we found that there is different types of fruit and colors. Also, we found that sometimes part of the flower remains with the fruit itself. There are some fruits which keep some parts of the flower. For example, the leaflets of the calyx. So some of the sepal are remaining into the fruit. After maturity, the leaflets of the calyx, for example, we found them into the pomegranate. We found that the leaf of the calyx, also the stamens, look at the picture and you will see that there is some answers are found inside the picture. The second part, or another type, which is the calyx may be take place as in eggplant and the date fruit. The caps which are found, the green leaves which are found into the eggplant here, they are part of the calyx here. The corolla leaflets, as in marrow fruit or marrow, we found them in this part. False fruit. The false fruit, it is a fruit in which the receptacle or any part share ovary to store food, as in apple, where the receptacle may share in fruit formation, or fig, or strawberry. So the receptacle here and the ovary together, they form the fruit, they fuses together and begins very swell with the fruit in order to form the fruit such as that of the apple or that of the fig or the strawberry. So we could summarize reproduction in the flowering plants by the pollination, providing the flower with male cell needed for fertilization. The fertilized ovule developed into the seeds. Fertilization also stimulates the auxins necessary for developing the ovary into a mature fruit, even if it is not fertilized. So that will lead us into another type of fruit, or another type which is called parthenocarpy. The parthenocarpy, it is the formation of the fruit from the flower without being fertilized. This can take place by two ways. Even natural parthenocarpy, as in banana and pineapple, or artificial parthenocarpy. This occurs by spraying. This is occur by the human, by men. So this artificial one, it is occurred by spraying the stigma with an extract of pollen grains. A pollen grains powder in ether solution, as indole or naphthol acid, acetic acid. So it stimulates the ovary to form the fruit. Then after this parthenocarpy, which is natural and artificial, is there another thing happen for this flower? We found that the flower, if it is not fertilized, we found that this mature flower, the fruit and the seeds often leads to stop the plant growth or leads to death, especially in annual plants due to the consumption of stored food. So we knew that the natural parthenocarpy and artificial parthenocarpy and the false fruit and how fertilization takes place and pollination and so on. But what will happen after that? For complete the story, we found that after the maturation of the fruit, the seeds often leads to stop the plant growth or leads to death, especially in annual plants, due to the consumption of stored food and inhibition of hormones. Pollination and fertilization do not take place. The flower withers and drops off without fruit formation. And now, my dear students, we reached the final point of our program. See you next time.